Swayam Prabha. Digital India. Educated India.
Welcome. Now we are discussing the synaptic plasticity, where we have discussed how the different kinds of modifications can take place, and now we will be discussing about the types of plasticity. In this course, for us the main point would be about plasticity that has to do with existing synapses, and that is what we will mean by that is synaptic plasticity, and that is that there is already a synapse between two neurons, and how the synaptic efficacy, the transmission of the synapse, efficacy of transmission, or the strength of the synapse, which is the current, that is the postsynaptic side's measure of the synaptic strength, that is changed with activity. So we have discussed that it is really the input and output activity that ultimately modifies the synapses, because at the synapse, if you remember, if we have the synapse, we will draw this diagram where we have the axon of the presynaptic terminal, and this is a spine on the postsynaptic side. This is the dendrite of the postsynaptic neuron, and this is the neuron cell body. This is the postsynaptic neuron cell body, and this is our synapse. This is dendrite. So if you remember, we are talking of how this existing synapses strength is changing. However, there are other types of plasticity where there are new synapses that are forming, new synapses between two neurons, or even the same neurons, and similarly, if certain inputs to neurons are not there, then the usually which neurons do not connect to those neurons, now they may be ending up creating projections onto those neurons. All these kind of things can happen. And similarly, here also there can be new spines forming, and collaterals may be forming, which effectively can increase the strength of these synapses. That is, due to the spike in the presynaptic side, there will be now more current in the dendrite of the postsynaptic neuron, and will effectively enhance the synaptic strength. So what are the types of plasticity? Then in that sense, first of all, we have what is called the structural plasticity. In structural plasticity, we essentially mean that there were no synapses originally, and new synapses are forming between two neurons. Also, in terms of structural plasticity, we can mention that initially there are lots of projections made by a set of axons from a neuron, from a structure going to making projections onto another structure, and with time, with activity, no activity in some of the synapses or many of the synapses, they get pruned away. And so that is also a kind of a form of structural plasticity, in the sense that the synapses that were made there that are not existing anymore. So, some of the examples of this kind of structural plasticity that have been studied is, for example, when a region of the cortex, is devoid of any inputs. I mean, the inputs to that region, are removed due to, let us say, a surgery or some kind of lesion, that the axons projecting to that region of the cortex. Let us say there are a bundle of axons that are going into that region, and I mean, it's necessarily not cortex, but for example, for the examples we will be talking about, it will be cortex. And there are neurons here, the large number of them, and this input is somehow gone. So, it is deafferented. So, afferents or the fibers projecting into that region are gone. And so, what happens in such cases, there are multiple examples, is that the surrounding regions actually start sending collaterals of axons, and finally that region is taken over by another region by including those neurons as their functional counterparts. And so, this whole region effectively starts to do the same sort of function. So, this kind of plasticity is structural and is also sometimes called as a expansion of maps, kind of plasticity. So, what there are multiple examples of these that have been studied, and for example, the primary example that we can give is that of representation of the digits in the somatosensory system. So, if you recall, when we discussed the sensory systems briefly, we said that in the somatosensory cortex, there is a map, in the sense that neurons at specific locations in the cortical region, in the somatosensory cortex, are sensitive to stimulation on particular regions of the skin or the body, and neighboring regions were mapped into neighboring regions in the cortex. Like in the visual system, it is called retinotopy. In the auditory system, it is called tonotopy. In the somatosensory system, it's somatotopy. And so, the hand and the palm and the different digits D1, D2, D3, D4, D5, they are represented nearby each other. So, in the cortical region, if we say that the neurons in this region are sensitive to D1, the first digit or finger, D2, D3, and so on. Now, the experiment that was done is that two of the fingers were bound together as if there was no function of any of a particular of them separately. So, whatever those fingers did were doing it simultaneously. So, in other words, the two fingers outputs, that the information that is being carried by the afferents finally into the somatosensory cortex of D1 and D2, or let us say D2 and D3, are now combined. They are turning out to be as if there is only one particular finger, and then the D2 and D3, this whole region became developed properties of stimulation on this entire fused digit. So, now the representation of D2 and D3 were not separate anymore. that is the previous D2 region could now respond to regions of D3, and previous D3 regions could now respond to regions of D2, and so they got combined. Similarly, when a finger is amputated in a case where it is a requirement, let us say, in that case the representation of the amputed digit. So, if you have the D2 and let us say D3, this is our D2 and this is our D3 region, then if the D3 region is cut off, then neurons from D2 and the neighboring D4 then start to project into the neurons of D3 and gradually take over parts of it. So, now since the actual inputs of D3 are totally gone, now the neighboring regions take up the neurons to function as a D2 or a D4 kind of representation. And so, this kind of plasticity is occurring throughout development, and also in such scenarios where in fact it is called the phantom limb effect, where a particular limb is cut off, then there is a feeling of presence of that limb, because the neurons that used to be sensitive to that particular limb or on that limb, regions on that limb, they are now being used by other regions of the body, and hence there is a sensation of that limb still being present, and that is only because of that region being taken over, the neurons that were representing that limb being taken over by the neighboring kind of positions on the body. So, this similar kind of work that has been done in the auditory system is basically just it's an expansion of a particular frequency, and this happens in a particular period of development called critical period. And that is a window of opportunity to modify the developing cortex permanently. And so, what is done in experiments, let us say if it is a mouse in which the experiment is done, or ferret, they have a specific window during their developmental period after birth, and if that animal is exposed to, so this is birth and this is the critical period, and then normal development and adulthood. So, manipulations done to the system in this period causes profound changes that are reflected in the adult. So, in the auditory systems, this map expansion has been shown to be like this, that if we present a particular frequency tone, let us say 5 kilohertz or 7 kilohertz tone that is presented during the critical period continuously, like tone and then a gap, then a tone, gap, tone, like that, it is presented continuously, then what happens is that in the adult, when you look at the tonotopic map in the primary auditory cortex, if you remember, there is low frequency side on one side and the high frequency representation on the side, and the medium frequencies in between. So, if we have regions of 2 kilohertz neurons that are sensitive to 2 kilohertz, then and gradually shifts to, let us say, 16 kilohertz or so in this region, that is, neurons here are tuned to low frequencies up to high frequencies on the other end. So, 7 kilohertz being somewhere in between, this is the actual usual area of representation of 7 kilohertz, in the sense that you get neurons with best frequency of 7 kilohertz. in that region, a majority of them. Now, when this 7 kilohertz is presented during the critical period, then this 7 kilohertz region gets an expanded representation in the cortex. That is, the regions that were for neighboring frequencies like the 6 kilohertz or the 8 or 10 kilohertz, those regions are now devoted to the same 7 kilohertz frequency. That is, in terms of the tonotopy, the representation of other frequencies neighboring to 7 kilohertz get reduced, while the 7 kilohertz expands at the cost of that reduction, and further away there are no changes. So, this again is a form of structural plasticity that is happening, that is caused by an activity during the critical period. So, similarly, in the visual system, there are cases where if you do if you stop representation, I mean if you stop stimulation of one particular eye, then the ocular dominance that we have talked about in our systems discussions, systems neuroscience discussions or sensory systems discussions, we have seen that there could be there are neurons in the visual cortex that get input from one eye more strongly than the other eye, and there are alternate bands of regions in the visual cortex, in the columns, which are dominated by inputs from one particular eye. So, that in the visual cortex we find the ocular dominance columns, in the sense that in the cortical region we have, this is the left eye dominated, this is the left eye dominated, this region is right eye dominated, and you have these alternate bands in the cortical region throughout the column. So, this is the surface and this is the white matter at the bottom, and the cortical column is along this axis, that is, and the layers are along this parallel to these lines throughout, layer 1, layer 2 to layer 6. So, in the normal case we get these ocular dominance columns where the neurons are majorly representing the one of the eye inputs, and they alternate. If during that critical period, if we do an eye suture, that is, one particular eye is devoid of any stimulation, then these ocular dominance columns do not form in the adult. So, it is only during that critical period that if you do the deprivation to one eye or stimulus deprivation to one eye, then the OD columns do not form, ocular dominance columns do not form. So, this is again another case of structural plasticity due to lack of activity of inputs from one particular eye, and hence that is translated across from the retina to the LGN, which is the thalamus of the visual system, and into the visual cortex, where these ocular dominance columns normally form. So, these are gone from the visual deprivation of one eye in the critical period. So, there are many more examples. I took only a few of the three systems to show you what this kind of plasticity is like. For us, it is going to be now a discussion mainly that we will follow, which has to do with the plasticity of synapses, that we will call synaptic plasticity, and we will be discussing this for the synapses that are already present, and they are being modified in terms of their strength or efficacy of transmission. So, and this synaptic plasticity that we will discuss occurs almost at all synapses in some way or the other, present in all synapses. Its presence may not be as we fall clearly as we will be discussing, but some form it exists almost in all of the synapses that we know. And this kind of changes in synaptic strength is ongoing and totally input-output activity driven. And we usually classify this synaptic plasticity in really two forms, that is a short term plasticity and long term. So, what we mean by short term plasticity is that let us say we measure the synaptic strength for a particular synapse by producing a spike in the presynaptic neuron, and we do averaging, and in the postsynaptic side we measure the membrane potential and the EPSP. So, let us say this is 1 millivolt EPSP that we see on average due to the spike in the presynaptic side. Now, with this with some kind of manipulation, again later on we can see that with the same measurement, that with the same spike in the presynaptic side, we may get either a decrease in synaptic strength, or we can get maybe an increase in synaptic strength, increase over the previous size, where the previous size is some, let us say, was like this. So, in the green, what we are showing, this is both, this is facilitation or potentiation, and in this case, in the case of the red one, where it is reduced, it is called depression. Now, the effect that we are seeing, if the system is left undisturbed, like you are not making any changes in the system for a while and do the same measurement again, if we get back our original synaptic strength that we had started with. So, if this postsynaptic potential EPSP is of amplitude A, at the end when we measure the synaptic strength again, we get back our A. So, this period of time where it recovers or it is left undisturbed is of the order of seconds to minutes. And so, the effect that we are seeing of depression or a facilitation, either of those, that is not long lasting, that is a temporary effect, and we regain our synapse back to the same strength. So, this temporary nature of synaptic plasticity is called short term plasticity, and particularly the two cases where we have short term facilitation with or short term potentiation, these are the two, which is our green case, and in the other case we have short term depression, which is in the red. So, this temporary phenomena of change in synaptic strength, that is what we are saying, because the EPSP is changing its amplitude. This is short term plasticity. On the other hand, what we mean by long term plasticity, which is long term, is that let us say again, we are measuring the synaptic strength by producing a spike presynaptically, and we measure the average strength of the EPSP. Again, let us say it's a millivolt height or a height of A. So this is the post synaptic side. Now again, we do some sort of manipulation to the activity of input and output, or just input, it depends. And then again we do the measurement, that we have a presynaptic spike, and postsynaptically we measure the postsynaptic potential. Let us say it increases to A prime. We leave it undisturbed for a while and measure it again, and we see that it is still A prime, or at least larger than A. It can decay a little, and it keeps going. You measure again, it's still A prime, and for measurable periods of time this remains at A prime or value larger than A, and saturates to that and saturates at that value. That is, it keeps on its synaptic strength. So this can be a period of hours, 1 to 2 hours, over which this kind of observations have been made, and this is what we will term long term plasticity, which has an early phase and a late phase. And we will be modeling a bit of the both short term plasticity and long term plasticity, at least the kinds that are used in general in models in the literature. And similarly, with that same other manipulation in this period, we can get a depression of synaptic strength. So this is, let us say, A double prime. So A double prime is less than A, and it continues to be so throughout that period of hour to 2 hours, and that is long term depression. So what we have here is long term potentiation in green, and we have long term depression here in red. So these are more permanent, more longer term changes in synaptic strength, and essentially it is not just the duration that is important, it is the temporary nature versus the longer term nature, and more specifically involvement of protein synthesis that shows that it is long term plasticity. Once we discuss more in detail about the long term plasticity, it will be clear when we will be modeling the processes of long term plasticity, then it will become more clearer as to what manipulation we are doing here, or what manipulation we will be doing here in order to observe long term facilitation and long term depression, or short term facilitation and short term depression. So next we will go on to more detailed discussions of short term plasticity and long term plasticity, and will be modeling each of them in order to implement them in networks which will be adaptive in nature, which will be learning based on activity. So next lectures will be on short term plasticity. Thank you.